I'm Suzanne Tough. I'm an Alberta Innovates Health Scholar in ACRI, the Alberta Children's Hospital Research Institute at the Cummings School of Medicine at the University of Calgary. Preterm birth is a big problem. It's one of the biggest problems in obstetric and neonatal care in Canada and the developing and developed world today. So we want to have as many children go to term, which is 40 weeks, as possible, because those children are healthier and stronger and more likely to thrive. Right now, we don't have a way to predict which women might deliver too soon and have babies that are too small. So women go through their pregnancy and we're not really sure which ones are going to make it to term and which ones might deliver too soon. So this test enables us to determine early in pregnancy which women we might want to watch more carefully. And the exciting thing about this analysis is that women don't have to have any symptoms of preterm birth. So it's one thing to help a woman once she goes into preterm labor, but the real opportunity is to identify which women might need more care early on or might need additional care or additional treatments. So this discovery really helps us identify early in pregnancy which women we would like to watch more closely.
I am Donna Slater. I'm an Associate Professor in the Alberta Children's Hospital Research Institute, Cummings School of Medicine, University of Calgary. Preterm birth is a huge problem worldwide, and one of the problems is that we don't have really good tools to identify those women at risk. And so what Dr. Heng and the team have done is we have developed tools to identify and predict women that are at greater risk of a preterm birth. This can be really helpful for clinicians in the future for development of personalised medicine and treatments for these women at risk. We collected maternal samples at two important time points during pregnancy, one at around 17 to 22 weeks of pregnancy when fetal ultrasound will be performed, and the other time point was later on when gestational diabetes tests would also be performed. And we took these bloods and we identified what we call a molecular snapshot of gene expression within these women at those times. The technique that we've developed is quite unique, and what we have managed to do is we've collected these bloods from the two time points, and we've collected them much earlier on in pregnancy. Most tests that are currently used to predict preterm labour are very late on in pregnancy. It's almost too late to be able to do too much about. By going much earlier, what we hope to do is identify the women much earlier, and this will give the doctor or clinician much more time and more opportunities to intervene and develop strategies for a prevention of a preterm birth, or in a worst case scenario, identification of a special care baby beds much earlier on. These are still very early days. We don't have a nice little kit that we can sort of use in the doctor's sort of office right at this moment, but the opportunity with this information to develop really good, strong predictive kits is very strong. I think it will happen with a lot of hard work and a lot of effort and collaboration with all of the scientists. I really see that we will get a predictive test within the future.
I feel super proud of Jan Heng for all the work that she did in this postdoc fellowship of hers, and I think it's a tremendous credit to her perseverance and to Steve Lai's perseverance as her mentor. These are very complicated analyses. They take time. We've repeated it, we've revised it. We've tried to make sure we've been as precise as possible, so I'm really excited from that perspective, and I feel like I'm thrilled from the All Our Babies cohort perspective that we could really contribute to such a complex question, and that women in our community have stepped forward and said, I'll be part of solving this problem. Like, I'll contribute my time and my energy to see if we can work through this. So I'm very grateful.